Hello everyone, I am Zuaib Mohammad Zia, Assistant Professor at PG and Research Department of Zoology in the New College in Chennai. So today we are going to have a brief look at a illustrative description of the working mechanism of PCR. PCR, as you know, is the polymerase chain reaction, which is used to amplify or make many copies of a small fragment of DNA. For an example, you imagine this is a fragment of DNA which you want to amplify, or you want to make many copies of this fragment of DNA. DNA, as you know, is a double-stranded structure. Here you see the two strands in orange color, and obviously it is anti-parallel in direction. That is, one strand runs in the direction 5' to 3'. The other strand runs anti-parallelly in the direction 5' to 3'. And you can also see the complementary base pairing. You suppose this is the sequence of this DNA. You have the complementary base pairs here, that is adenine pairing with thymine, cytosine pairing with guanine, and these lines what you see here are the hydrogen bonds in between the two strands. Now moving on to the working mechanism, it primarily consists of three steps. The first step being denaturation, which operates at around 95 to 96 degrees. The first step involves breaking the bonds in between the two strands. So you can see here there were bonds between these two strands. In the first step, we have broken the bonds between these two strands, and we have separated the DNA fragment into two single strands. So from a single double strand, it has become two single strands. Now the next step which comes into play is the primer annealing. Primers are nothing but short oligonucleotide sequences. You can see here these blue sequences. These primers are designed specifically such that they are complementary to the 3' end. So this is a 3' end of the DNA fragment, and this is the sequence AATT. You just design a complementary sequence which reads again T complement T complement A complement T, and A complement T. You design a sequence in such a way that it has complementary base pairing, and thus it can bind. You design one primer for this 3' end. Similarly, you design one primer for this 3' end. So for every fragment, you have to create two primers. One primer, this one, it will be called the reverse primer, and one primer, this one, it will be called the forward primer. So again, this step is called primer annealing, which operates around 66 or 68 degrees Celsius. Then the final step comes in, which is the primer extension, which takes place because of the polymerase enzyme. For this PCR, you must be knowing the most common enzyme, the most common polymerase enzyme used in PCR is the Taq polymerase, TAQ, extracted from Thermus aquaticus bacteria. It is a thermostable polymerase enzyme. So in this step called primer extension, the polymerase enzyme extends the primer. This primer gets extended in this direction. Thus, called as a reverse primer, and this primer goes extending in this direction, and thus it is called the forward primer. And these three steps that we have seen, this denaturation, primer annealing, and primer extension, constitute one cycle of the PCR, and many such cycles are repeated, and moreover many such fragments. What we are seeing here in this illustration is just one fragment. Many such identical fragments are extended for multiple cycles, giving millions and millions of copies of DNA. So these you can see the original strands. The orange color ones are the original strands. The blue color ones are the new created strands. This is one cycle. And the next cycle again it gets doubled. Next cycle it gets doubled, and so on, giving millions and millions of copies.